Okay, so I'm going to take this prokaryotic transcription lecture because I didn't do that. Let's go. So this slide is the little bullet point summary of transcription. So it is using DNA as a template off of the dedicated template strand to make a transcription, an RNA copy. The role of the RNA can vary, as we're going to see, particularly in, well, in all, we need tRNAs, we need rRNAs, and we need messenger RNAs. But they all start from DNA. And let's go to the next slide. So the convention, as we've been talking about in manipulating DNA so far, is to consider that we generally orient ourselves by saying the top strand is going from the 5' to 3' direction, which is defined on the orientation of the bonding, right? So the 5' phosphate is sticking out here, and it connects with the 3' hydroxyl on the next one to get 5' to 3' bond like this. Well, even that's very convenient for a lot of reasons, but it's important to realize that template strand is actually the other strand, the strand on the bottom there, which is, that's how it goes. So, but as a result, the top strand, the one that we refer to a lot, actually does represent what the RNA looks like. It represents the product of transcription, in that here with the template strand on the bottom of this DNA duplex, you can see what happens if you transcribe it and turn it into RNA, and that the sequence becomes the complement of what's there, with the exception that wherever there an A is instead of adding a thymine opposite it, we've added uracils. I think everybody's familiar with that from the beginning. So, but if you go ahead and actually look at the actual sequence, with the exception of the T's being changed to U's, this top strand, the one that we orient ourselves on in terms of designing genetic constructs, also represents the coding sequence. So you could actually look in here and translate this into codons. And so that's that. And you can see that both in this sort of nice typed up picture or the sort of drawn out picture here if you need to see it. So we have the DNA transcript melted here as your duplex. This region here showing 5' to 3' on top, and the 5' to 3' goes this way, so our 3' is over here. Remember, the polymerases are adding on to the free 3' end, a loose 3' end there. That's the part of that's one of the substrates. And then, but this coding strand, the template strand down here on the bottom, then is the template and generates this transcript. Okay, so this is just a quick look at what bacterial polymerase looks like and eukaryotic RNA polymerases look like. I'm not going to get a lot into that like we did with DNA polymerase. I mean, they have I think a crudely similar structure. They have an opening in the middle that has their active site. It's described sometimes as a crab claw as opposed to a hand, but I think that's that. They have a fundamental similar mechanism with different binding pockets, and they both DNA polymerases and RNA polymerases require magnesium for function. So there's some fundamental similarities in their biochemistry as well. Magnesium, with the obvious exception that they work off of different templates. On its own, RNA polymerase does not encode an exonuclease proofreading mechanism either. So that's a unique function that's encoded in DNA polymerases. This slide just kind of summarizes the basic steps that are involved in the initiation of transcription. So you're going to start off with DNA template, which is shown at the top here. Within it is a dedicated sequence that the transcription needs to start at, and this is called a promoter. And these are defined by certain sequence elements. And in the absence of a promoter, RNA transcription won't happen. It binds not just the polymerase itself, but some proteins that facilitate the binding of the polymerase. And that's it. We'll see that example in spades when we get to the eukaryotic system. But in any case, the RNA polymerase binds to the DNA promoter, and at first it's kind of wrapped around it. As you can see out here, it's not, this bubble here represents the polymerase, the kind of structure here, like the claw/hand thing here. And here's the DNA still wrapped around the outside of the polymerase, and that's described as a closed complex. In order for transcription to occur, this DNA needs to melt here. You need a little localized melting or unwinding of the DNA. Promoter sequences do tend to be or be very closely associated with AT-rich regions. And then the template strand can get dragged into the catalytic region of the polymerase. So now you've gone from a closed complex to an open complex, and then initiation happens, which is the first actual copying of an RNA off the template. And after you've had the first few nucleotides are added, then you transition, you go from open complex initiation, then you have what's called an elongation complex here. And so you can see that the double-stranded DNA comes in, it gets melted, and the RNA is protruded out the back end here, and the duplex is reassembled on the other side of the polymerase. When you get to the end of the gene, the RNA polymerase will dissociate, it'll release the finished transcript, and it can go, it's an enzyme, so it can keep working. We're going to look at examples of regulation, both around step 1 here, which is this recognition and of the promoter. We're also going to look at an example of elongation regulation, which is around here, because obviously gene expression needs to be regulated. So before we get on to the regulation and some of the protein specifics, let's just take a closer look at this in vivo. So here is this similar picture that we just looked at, kind of blown up. So again, you have your RNA polymerase showing here. Double-stranded DNA helix is here. Because you're unwinding here, you have a little bit of overwound DNA in front and a little bit of underwound DNA in back, not just kind of like with replication. It was of a twisted molecule, so we're accentuating these various twists. But more importantly, what I want to show you is on the top part of this slide, in that you can see here, if you look carefully, you can see the thin line. This is an EM, and this shows the gene, and this is where the transcription start site is, right here. And so these little bumps are transcripts. They're not just transcripts. As the ribosome goes along, as the RNA polymerase moves along here, it's making the mRNA, and then when there's enough mRNA assembled, when there's enough mRNA transcribed, then ribosomes can assemble on top of it as it's being transcribed. So you have linked transcription and translation in prokaryotic cells. And so what you see here is as the RNA transcript gets longer, so too do the number of ribosomes that are bound to it. And so this transcript is actively being translated into protein as it's being made. So they're coupled. Transcription and translation are coupled. So now I want to start talking about the promoter elements of genes. And so the first thing we're going to talk about is the fact that, well, all genes have a promoter element. And here's a basic prokaryotic promoter element. So we talk about the promoter element in reference to where transcription initiates. So here is this +1 site indicating the first spot where the transcript initiates. And 10 nucleotides upstream from that, we have this AT-rich region. And down on this copy here, so this is our start codon, and maybe the transcript starts just a little in front of it, then we can see the an AT-rich region right here. So it would be TAA, TAAA, or TAAT. So that's probably the -10 TA-rich region. We're going to see another TA-rich region in eukaryotic promoters as well. And then farther upstream from there is another region, It's called the -35 region, and that is cropped off of this one. And then even farther upstream from there, there's a proximal promoter element that is a region that tends to be AT rich, which helps with melting, local melting and destabilization of the DNA structure. Now, it turns out that these promoter elements and their interactions with polymerase are sequence specific. So we're starting to look at some sequence specific interactions of DNA with protein, kind of like, well, we've seen some before, restriction enzymes. It's definitely determined by the sequence specificity of the restriction enzyme for its recognition sequence, and then the protein interacts with the DNA to cut. In this case, protein interacts and just binds and then actually recruits other proteins to do other catalytic jobs. And so this is an example here of looking at the promoter for the lac operon, which we're kind of familiar with from lab a little bit, I hope. So here's the consensus promoter elements that I just told you about, but you can see that actually the wild type lac operon is not a perfect match to the consensus. So instead of going TAAT, this one goes TAT, GTT. As a result, if you were to test this in an in vitro assay for transcription activity, you would see that the wild type lac promoter has less activity than the consensus sequence, which is equivalent to, let's see which one, this number, mutant 2. It looks like that's the one that matches the consensus sequence. So that's fine. Not all genes need to be expressed at the maximal amount. Each gene needs to be expressed at the amount appropriate for that gene in the biological situation it is responding to. But we can see also is that if we put in a mutation, let's see which is the first mutation, is they put a deletion in up here, and it actually works a little better. They made the mutant look exactly like the consensus, and that works much better. If we have the consensus sequence here and the deletion, we get even better. That's optimal, so that's sort of like the optimal promoter right there relative to the wild type lac promoter. And then if we can add another mutation down here in this other element here and lose a little activity. But this just shows you a consensus sequence. It means it represents the commonalities of DNA sequences over many, many different samples. And so there are many cases where a promoter element or something doesn't match perfectly to the consensus, but it's still considered functional. And the other thing to note about this is that the promoter activity is determined by the sequence in the promoter binding sites, as well as the spacing is important, which suggests that there's a very precise physical interaction going on between the binding of the DNA, let's do one strand for simplicity, and the RNA polymerase interacting in some, precise manner. Now I want to talk a little bit more about the lac operon for a second, because it brings us to our first example of regulation. And this is actually regulation, not so much, it's actually kind of a double example of regulation. But if you take a look at the lac operon, you may remember that the lac operon is activated only in the presence of lactose. When there's lactose in the bacteria's environment, it needs to turn on these genes, which allow the bacteria to digest lactose. Normally, it's digesting glucose. So the way this is regulated is that the genes are usually in their off state, which is represented in this picture here. So what makes them be in their off state? Well, there's a regulatory protein that's separate from RNA polymerase. It's called LAC-I. It's a LAC inhibitor protein. It's actually made nearby. And LAC-I binds with other sequences near the promoter. And so you can see what happens is LAC-I is bound here. And it's bound on some of these operator sites. In this case, it's bind on operator. There's a couple options for it to bind. So it takes 2 operator sequences for it to bind fully. And so it can either bind here in here, and this is its preferred binding location. And it being bound there, now when an RNA polymerase is assembled on the promoter, they can't get past this structure. So there's a promoter upstream in this region that could make a transcript, and they'll just keep going. It would keep going through, and it can't get through this structure where the operators are looped together and bound up with the repressor. And on top of that, there's a second promoter element that's actually being fully blocked from even interacting in a functional way with polymerase. So one polymerase is being blocked from interacting with, one promoter is being blocked from interacting with polymerase, and the other promoter, even though it's engaged with polymerase, it can't make a functional complete transcript. So actually seeing a little regulation at this initiation stage and at the elongation stage. And the example on the bottom just shows that if instead the lac repressor protein binds with the other two other of the operator sequences shown here, both promoters are exposed, but LAC-Z is now inaccessible, and more importantly, polymerase can't pass through this big lump, blocked from passing through. But as soon as lactose is detected in the environment, let's put some lactose in here. Then the repressor protein dissociates, which means it's going to come off. So now it'll be floating nearby. Here's our repressor protein. Now it's dissociated because it's bound with lactose. And now the polymerase can get through just peachy. So now it can get all the way through. And so that's an example of how a gene is regulated. Both, and I said this actually gives an example both by blocking initiation on the promoter and also blocking elongation off of an adjacent promoter. Now, in that case, we were just talking about, I guess, the most generic sigma factor in prokaryotes, and we showed that it interacts with this -10 sequence, this -35 in this, and then with this upstream AT-rich region. But there's actually more than one sigma factor in bacteria, and this is a more global form of regulation, and we're going to look at those on the next slide. So here we can see a variety of sigma factors. So this is the kind of standard one we just actually looked at with the -35 sequence and this TAT-like box, TAT. You can see there's some alternate sigma factors that can mediate the interaction between the genes and RNA polymerase. And so there's sigma S, sigma F, sigma H, and E, or they're designated by their sizes here, 38. I think the sizes are 28 or 32 or 34. And so you can see each sigma factor is actually associated with different motifs, still in approximately the same locations in the promoter, but the sequence specificity is different. So we're seeing that these different proteins have different sequence specificities. And each of those specificities is associated with a subset of genes. So in this way, the bacteria, when it recognizes certain changes in its environment, it can activate a set of genes and help it respond. So heat shock is a very common stressor for bacteria, and it's a universal stress response, but it extends through all organisms. And so you can see here there's a specific sigma factor and a specific consensus sequence in the promoters with both -10 and -35 that dictate and drive the heat shock response. And then you can see that for these other, starvation has its own response. Flagellar structure and movement has its own response, which you can imagine the need for that. And then also extra cytoplasmic stress, which just might be any other type of stress, could be from changes in pH, salt conditions, that are distinct from heat or food. But the main point here is that there's different proteins, different specificities, and are targeting different promoter elements to create specific patterns of gene expression. So I want to talk about a technique here that's used first in E. coli, and then we're going to see it again when we look at some other transcription factors, and it's called DNA footprinting. So how does this work? It's all smushed up on this slide, so I'm going to move ahead to the next slide where the images are a little larger so we can take a closer look at it. So basically, you start with a piece of DNA that you suspect or know that can bind a protein. So here it's this promoter element, okay? And it's a finite piece, and maybe 80, less than 100 base pairs long. And someplace in it, your protein can interact. And what you do is you have a radioactive signal just at one end, which is shown here by the red dot. And then you set up two reactions. In one reaction, you've added your protein to the radio-labeled DNA. In the other reaction, you don't add any protein. And then you treat either mixture you treat with DNase 1 at a carefully controlled concentration. So you just get just a little bit of cutting. So throughout the population of DNA molecules where it's unbound to the protein, you wind up with a population representing every nucleotide in the ladder, because each one got cut once, and randomly you eventually cut at every different spot. In contrast, on this side, if you have a protein bound, it blocks the cutting by the DNase. So you're not going to see every single location in your DNA ladder. You're going to have places that were blocked because the binding of this protein interfered with the ability of DNase to cause degradation. And then you run that out on a gel. So this, before we move ahead, I'll just show it again, and their drawing's a little neater than mine. Where there's no protein bound, you get random cutting representing every different place. There's a nucleotide. Every different place there's a substrate for DNase. But where there's a protein bound, it blocks the DNase from cutting. And so that will let you footprint or map. There's our protein right here. This lets you map where the protein blocked the catalytic activity of the DNase. And then you can go ahead and run that on a gel, which is shown here. And so we see a couple things in this gel. Here's first just a control where the DNA was just treated to the DNase, so we can see what the ladder looks like on its own. And you run it out, and you can see a spot representing every nucleotide. This gel isn't sequence specific, but if you already know the sequence and you know down here it was an A and this was a C, just because you knew the sequence, you would still be able to map what sites are being blocked by cutting. And there are more advanced versions of this technique where you actually do this in sequencing-like reactions as well. And then these two columns here, this shows what happens if you bind sigma factor 38 in the location where it binds. So when protein was bound, it blocked cutting by DNase. But that's a sequence-specific phenomenon. So if you put in a different sigma factor, there's no protection in that region. Okay, so now we're going to change gears and talk about termination. There are two types of termination I'm going to talk about that happen in E. coli. One's called rho-dependent and one's called rho-independent. So once again, here is our elongation complex whizzing along, producing its RNA at the back end here. At some point, it's going to get to the end of a gene and it needs to stop. So what are the signals that tell the RNA polymerase to stop? The first one is called rho-independent, that I'm going to talk about. And that termination is actually controlled by sequences within the gene, because rho is a protein. So that means this is a protein independent. Not only is it rho independent, but there's no other protein involved in it. So rho isn't required for this termination, and it's just simply terminated by the structure that's formed here. So the polymerase actually is transcribing along here. It gets to this termination region, which has got a dedicated structure that's going to form it. We'll take a closer look at that in a second. It forms a bit of a hairpin. And then once the hairpin forms and the polymerase has moved past it, the hairpin causes the polymerase to dislodge. So let's just take a little closer look at that. So you can see here that if there's some inverted repeats in the DNA sequence. And so as this is the coding strand down here, it's going to go along, and then there's a little bit GC rich right in here, and they're going to bind up to form this hairpin. That'll be relatively stable structure because it's GC rich. A little loop at the end of U's, and then it also has some more A's which get transcribed to U's just past the loop. And this element, when it interacts with the polymerase, it causes the polymerase to dislodge, and that transcription, that gene is done, and then the polymerase can scoot back to the beginning and start again, or start on another gene. This is just another look at the same thing. I thought it was a nice kind of colorful and cartoon drawing. This row independent mechanism is also called a type 1 terminator. And it's the same thing is depicted here, and less sequence and some less specificity in some of the details, but the RNA passes through, we get this hairpin loop, and then here's your completed transcript with that terminator sequence at the end. Okay. Now we're going to look at row-dependent termination. And what is row-dependent termination dependent on? It's dependent on the protein row, which is, you can see it there, which binds to these sequences also in the gene. They're called RUT sequences. And again, the polymerase actually has to transcribe through the RUT sequences. And once they're transcribed through, then the row protein binds to the transcript, the native transcript, and by the structure, this is the image from your textbook, this is what they use to represent a helicase generally, and it kind of can whiz along there and scoot up and twist along the transcript and get up to the, and it is an ATP-dependent mechanism and causes disruption of the dislodging of the polymerase from the template. And so that is, again, row-dependent termination. So this requires the protein row requires a RUT sequence. And again, in both cases of these termination events, the polymerase has actually gone past the signal that triggers the termination, which then kind of feeds back on the polymerase and tells it to stop. One's mediated by a protein and one is not. So here's a summary of the stuff we talked about in this section. So we talked about the orientation of the strands, the relative locations of promoters, the 5 basic steps in the transcription cycle. Sigma proteins is an example of proteins that are involved in regulating the initiation of transcription, and that they're tissue specific, so they're our first taste of transcription factors. We revisited the mechanism of the lac operon. We also looked at termination in this context of this row-dependent or protein-dependent mechanism and an protein-independent mechanism of termination. And we also looked at DNA footprinting, and again, I believe we'll be coming back to that again in context of some eukaryotes and some experiments that we're going to look at by the end of next week.